Okay, we are going to move into Chapter 3, and you will probably be relieved to know that Chapter 3 is not quite as long as Chapter 2, so they're not going to be 8 installments of Chapter 3. They're really just going to be 3 of them, so hopefully that will be a lot easier to deal with. We're going to start out talking about the structure of the synapse. So synapses are specialized points of contact between a neuron and another cell, whether it's a neuron, a muscle cell, or a gland. It's the point at which information is transmitted from the neuron to the postsynaptic cell. So the postsynaptic cell can be a lot of different things, but that is our point of contact with the axon terminal. So and just in terms of terminology, which of course I want you to know all this terminology, the presynaptic terminal is formed by the axon end projections of the cell that's transmitting the signal. Okay, the postsynaptic terminal is formed by the membrane of the receiving cell, be it another neuron, a muscle cell, or a gland. The synaptic cleft is the space between the two terminals. Okay. Neurotransmitters are the chemicals that are contained in vesicles in this presynaptic terminal, and those neurotransmitters are used to transmit information across the cleft by binding to a receptor in the postsynaptic membrane. So there's a really nifty little electron micrograph of a synapse here. And you can see in the presynaptic terminal, you can see those little bubbles that are the vesicles of neurotransmitter waiting to be released. There's the synaptic cleft between the two cells and the postsynaptic membrane on the other side. So that's a pretty cool picture that you can actually see that. It has a scale here, 0.25 micrometers or micrometers. That's pretty dang small. So there it is. Hey, there's a little 1-minute video animation of the synapse if you want to watch it, and then the Australian guy, Tutor Tom, with a 15-minute explanation of electrical and chemical synapses, which you can watch or not at your discretion. I talked a little bit in Chapter 2 about the different types of synapses. We will refer to an axosomatic synapse, which is where an axon synapses with the body of another neuron, an axodendritic synapse, where the axon synapses with the dendrite of another neuron, and an axonic synapse, where the axon synapses with the axon of another neuron. So those are 3 different. And then there's some specialized synapses like the neuromuscular junction, which is its own little specialized thing. Okay, so we will begin talking about the events at the synapse in just a moment. So on page 49 in your text, there is a list numbered 1 through 7. I want you to know that list. Okay, and we're going to go through them one by one. It's pretty simple, hopefully, straightforward. And one of the assignments for this chapter is that I actually want you to describe those 7 steps in your own words, not using any of the terminology, like the action potential and the membrane. So you can do it however you want. You can use an analogy. You can use sock puppets and record yourself doing it and post the video. You can do it however you want, as long as you describe the events at the synapse, you include everything, and you use your own words. Okay, so there's a description of that in the assignment and which words you're not supposed to use. Okay, so we're going to go through one by one. You can follow along here on the PowerPoint. You can follow along on page 49 in the text, or you can follow along in Canvas in that content page named Events at the Synapse. Okay, so the first thing that happens at the synapse, and there's also figure 3.2 in the book on page 50, it shows all these steps. Little picture for each step. Okay, so number 1, the action potential arrives at the presynaptic terminal. Okay, that's the first thing that happens. Second thing that happens, the membrane of the presynaptic terminal depolarizes, opening calcium channels. Okay, the third thing that happens is calcium flows into the terminal. It also liberates calcium from intercellular stores, and that calcium influx triggers the movement of synaptic vesicles towards a release site in the membrane. So when you look at this little diagram, the one of the synaptic vessels has fused with the membrane and it's released its neurotransmitter into the cleft. The other ones are waiting there, but you can almost imagine that they're moving towards that release site. Okay, so they move towards the release site, and the fourth step is the vesicles fuse with the membrane and release their neurotransmitter into the cleft. Okay. The fifth step is the neurotransmitter diffuses across the cleft. Makes sense, right? The sixth step, any neurotransmitter that contacts a receptor on the postsynaptic membrane binds to that receptor. And the seventh step is that the receptor changes shape. Okay? So changing the shape of the receptor can do a couple of different things. It can open an ion channel that's associated with that membrane receptor, like we talked about in Chapter 2, or it can activate intracellular messengers that are associated with that membrane receptor. So this is often referred to as a second messenger syndrome. I'm sorry, a second messenger system. And we are not going to go into the detail that there is in the book about the second messenger system. We're just going to say that it's a possible thing that could happen when the neurotransmitter binds the receptor. Okay, so I'm just going to review that one more time. Number one, action potential arrives at the presynaptic terminal. So in the diagram in the book on page 50, which is right here, the action potential is represented by that big black arrow. Okay? So the action potential arrives at the presynaptic terminal. The second thing that happens is calcium channels open because the terminal was depolarized. Okay, so in the second picture down here, calcium is rushing into the cell. The third thing that happens is the vesicles, the synaptic vesicles, move towards a release site. They're triggered by the movement of calcium. So here they are on the edge of the terminal, waiting to be released. The fourth thing that happens is those vesicles fuse with the cell and release their neurotransmitter. Okay. Fifth thing that happens, the neurotransmitter diffuses across the synaptic cleft. Then it binds to the postsynaptic membrane receptor, and the membrane channel changes shape, triggering either the opening of ion channels, which could generate a local potential, and if there are enough of them, it'll generate an action potential, or it could activate intracellular messengers associated with that membrane receptor and start a different cascade of events. Okay, so those 7 steps are the ones you need to know. Okay, we're going to wrap up this recording, and in the next section we will talk about electrical potentials at synapses.